pleasure to introduce our guest speaker, Dr. Paula Bates, a U of L faculty member for over 20 years. Dr. Bates earned her degree in chemistry from Oxford University, then pursued her PhD in biophysics at the University of London, followed by a postdoctoral fellowship at the University of Alabama in Birmingham. In addition to serving as a faculty member in the Department of Medicine, she is also an accomplished researcher and has published in many professional journals. She also serves as the director for the Excite Hub, one of 3 sites in the U.S. focused on increasing the number of university technologies into real-world products. She has overseen over 150 of these technologies that address a wide range of health-related problems. Dr. Bates is a member of the team of U of L researchers that have discovered a technology that is believed to block the coronavirus from infecting human cells. U of L is in the process of a fast-track development and seeking approval from the FDA to start treating patients that are seriously affected by the COVID virus. Please join me in welcoming Dr. Paula Bates as our featured guest. We're anxious to hear more about your work.
Thank you. And let me just get set up to share my screen here. Hopefully you'll see it. Let me see. Okay. And there you go. Can you all see that now?
Can.
Okay, well, thank you, and good afternoon to everybody. It's a great honor to be able to talk to you. I thought long and hard about what I would talk about today because my coronavirus research is a rather recent thing. Actually, I'm a cancer researcher by training. And so I thought I'd kind of tell you my story. I'm sure probably a lot of you are not familiar with what scientists do, and my story strangely owes a lot to serendipity. So I thought that I would make that the kind of central theme of my talk today. So first of all, let me define what I think of as serendipity. There are lots of different definitions, but generally it's finding useful things by accident. And the one that I think applies both to me is this one at the bottom here, where in many cases I've been looking for something, I've found something else, and I've realized that what I found was probably more useful than what I was looking for in the first place. And so I think you'll see that that is a theme that has followed me throughout my career. Just to backtrack a little, I'll tell you a little bit about me. I think of myself first and foremost as a researcher. I run a lab that does studies the biology of cancer cells, particularly trying to figure out how cancer cells differ from healthy cells so that we can target them with new drugs that should be specific for cancer cells. I've had a very translational focus throughout my research career. In other words, I try to take whatever I discover in the lab and turn it not only into scientific papers, but into something practical and useful, for example, a cancer drug. As part of my kind of research endeavors and translational endeavors, I've been a mentor to many students and young trainees. I'm an inventor on several patents. I've founded a couple of companies here in Louisville. I've collaborated with industry, and as mentioned, I've directed programs that teach other people how to do those kind of things. So what do I actually do all day? Well, I supervise a research lab. I'm not the one, only in press photographs am I the one really wearing the lab coats. These days I'm mostly in my office when it's not Corona time at least, writing grant applications, trying to get support for the research that we do. And the other thing I do is these 2 actual translation and commercialization programs. Excite was at the University of Louisville. And Kinetic is a new effort, which is actually statewide. It's a collaboration between the University of Kentucky and the University of Louisville and all of the actual public health universities in Kentucky. I'm British, in case you can't tell from my rather strange accent. I've been here for 20-something years now, so it's gotten a little mixed up with some Alabamian and some Kentuckian in there. I went to school in a small town called Nantwich, and you've heard about the rest, Oxford, and then London, and then Birmingham, Alabama. I've been in Louisville actually since 1999, so I consider Louisville my home. I'm very saddened, as is everyone, by the events of the recent days, and hope that we'll see some more peace soon. But I consider this home. I moved here right after my fellowship and settled down here, have a family here, and I say, consider it home. So back to serendipity, I told you this kind of idea of looking for something, finding something else, and deciding that was better than what I was looking for has been a theme for me. And that is true in my career too. I didn't start off wanting to be a scientist. I actually wanted to be a police detective. I think I'd read too many Famous Five or Nancy Drew mysteries or something. But when I was very young, I wanted to be a police detective because I liked solving mysteries. But in Britain at the time, there was a height restriction. You had to be at least 5'2" to be a police officer. And so by the time I was about 12 or 13, it became abundantly clear to everybody. If I was here in person, you'd see that I'm less than 5 feet tall. It became clear that I was not going to be tall enough to actually be a police officer in England. So I decided at that point I would be an engineer. And unfortunately, at that time in the 1980s, I was told that they didn't have the facilities for girls to come and work on the factory floor of Rolls-Royce, which was my local engineering factory. So they sent me to work in a hair salon instead. And as you can see by these photos, that had a very traumatizing effect on my hairstyle choices for the next several years. I then decided to be a pathologist, but realized very quickly that I'm extremely squeamish, and I'm one of these people who passes out at the sight of blood. So that was not on the cards anymore. And then when I finally got to college, I was studying chemistry. I liked chemistry. I decided to do a project involving lasers, but once again, my height got in the way, surprisingly. I was actually too short to reach the stands where the lasers were kept. And so I was told that I couldn't work in that lab anymore. And it turned out to be a fortunate thing for me. I went to my second choice lab, which was a woman called Alison Roger, who's been a wonderful mentor throughout my career, who did cancer drug discovery. And I found out that was something that got me really excited, and it's what I've spent the rest of my career doing. When I'm not doing science, just in case you have this vision of scientists as spending all their time in the lab, I try to do other things. I'm married with 2 teenage children and a very pesky dog who so far is behaving himself, thankfully. My husband's from New Zealand, I'm from England, so we do a lot of traveling between continents, at least when it's not Corona time. And in any spare time that I happen to have, I mostly revolves around food and drink. And working out to try and mitigate the effects of all that eating and drinking. So now on to the research part. I'm not going to tell you about everything I do. I just wanted to highlight a few things. And again, the kind of defining factor is a lot of these things, there's an element of luck involved in how I discovered them. The first one is the one I'm going to talk about most, which is this aptamer called AS1411. It binds to a protein called nucleolin. It forms a strange structure that you see here, and it's able to kill cancer cells. And also we've taken it into clinical trials and find it's also able in at least a few patients to shrink their tumors. I'm working on a kind of next generation version of that that's attached to nanoparticles, which should make it work even better. And then the other part of my studies are various different things that I've discovered by chance. And the focus has been trying to find things that are selective for cancer cells, but don't harm normal cells. And so let me get back to this Aptamer. Aptamer is a strange kind of in-between drug. When we have cancer therapies, we have these small molecules such as chemotherapies, and we have these large monoclonal antibodies. And the Aptamers lie somewhere in between, and they're made of DNA. So they have some of the advantages of both classes. As I mentioned, serendipity has played a large part. Actually, this was discovered completely by accident many years ago. Again, you can see how old I was there, but many years ago, we were studying one thing. We were trying to develop DNAs that target specific genes in cancer cells. And we added them to the cells and came back the next day to see what happened, and found that some of the sequences of DNA were able to kill cancer cells very effectively. However, they weren't the sequences that were supposed to kill the DNA. They were other control sequences that weren't supposed to do anything. And so we quickly realized that, well, this isn't working the way we thought it was working, but nonetheless, it's very interesting. Is it specific for cancer cells? How does it work? And is this something we can develop as a useful therapy for cancer? And so that's what we did. We followed this up. We showed that it was active against many other types of cancer cells, and that it had much more effect on cancer cells than on normal cells. And what you see here is just pictures of cells growing in a dish. And when you treat cancer cells with the Aptamer, they all disappear because they've died. Whereas the normal cells look just almost exactly the same because they are very healthy. It turns out that this works by binding to this protein called nucleolin. I'll tell you a little bit more about that later, but it turns out that it's present on the surface of cancer cells, but not on normal cells. And this picture here just shows you in green the presence of nucleolin on those cancer cells, but not the normal cells. And this protein kind of helps cancer cells survive, at least, especially when it's on the cell surface. And so binding to that and blocking its function is able to kill the cancer cells selectively. My colleagues, John Trent, Don Miller, and I took this into formed a company called Aptamer, took it into clinical trials, and it's been tested in over 100 cancer patients. What those clinical trials showed is that this drug seems to be very safe. There were no serious side effects associated with treatment from the drug. And in a handful of patients, there were some very dramatic clinical responses. This is a gentleman who had a metastatic kidney cancer who had a very nice response. His cancer went away, and he stayed alive for many years after. But the limitations of the drug is that it's quickly cleared from the body. And again, we don't know why, but we know that a handful of people have very good responses, but most people do not. And so we're still working on this. We're trying to identify ways to select those patients who might be most likely to have these great responses. And as I've mentioned in the past, we're also generating new versions of AS1411 that will circulate in the body more and hopefully will be more effective in the general population. And so you may be wondering what on earth this woman who's supposed to be talking to you about coronavirus is going on about cancer for so long for. And that's because it's this research in cancer that led me to coronavirus. And so back in late January, I started to be watching the news coming from China and seeing that there was this new virus that some people were starting to get worried about, and realized the potential of this to have very kind of severe and widespread effects. And unfortunately, this came to pass. And so here we are dealing with now over 100,000 deaths in this country alone. But it was my knowledge of the previous research related to nucleolin that gave me an idea about how to try to tackle this new coronavirus. And the official name for that virus is SARS-CoV-2. And so again, I just wanted to remind you about nucleolin, that in normal healthy cells, either there's not very much nucleolin there, or it's on the very inside of the cell in these structures called the nucleus, and that kind of darker dot is called the nucleolus. But we know that in some cells, when they get stressed or when they're subjected to certain signals, the nucleolin comes out of the center of the cell and goes into this bag of fluid called the cytoplasm and to the surface of the cell. And in the cancer cell, there's a ton of it on the surface and outside of the nucleus too, and all over the cell. And so that's how it works with the cancer selectivity. When you're a piece of DNA floating around on the outside of the cell, all you can see is what's on the outside. So you see the cancer cells, but not the normal cells. But the other thing that Aptamer can see is cells that are infected with virus. So the other thing we know about this protein called nucleolin is that it plays a role in the activity of some viruses. And so what you see on the right there is a whole list of different viruses where nucleolin has been implicated in having a role. And the ones highlighted in yellow are are actual other coronaviruses. And so in this role varies. In some cases, it's just been shown that when the virus binds to cells, you see this nucleolin transfer to the cell surface. And in other cases, it's been kind of more defined what role it plays. Sometimes it helps the virus get into the cell. Sometimes it moves the virus around in the cell. And other times it helps the virus replicate inside the cell. And so my idea was very simple, based on the fact that we know that this protein nucleolin has a role in a lot of viruses, and we have something that can block the function of this protein. Can we use the nucleolin aptamer to block SARS-CoV-2, the new coronavirus? And so, you know, if I'd been anywhere else in the country, or most other places in the country, the idea would have stopped there. It would have stayed just an idea that I probably would never have gotten to test, or it would have taken a very long time to test it. But fortunately, I'm in Louisville, and I'm not sure if you're aware, but we have really pretty amazing facilities here for doing lots of different kinds of research, but especially research on emerging pathogens such as the new coronavirus. We are one of 12 regional and 2 national regional biocontainment labs. And part of our Center of Predictive Medicine, which is run by Kenneth Palmer, which is who you see here. And they are able to do, they have the high kind of safety standards to be able to do research with this dangerous virus. And so I talked to Kenneth and told him my idea. And fortunately for me, I got in pretty early back in February, when he wasn't yet swamped with all these requests with other people who had similarly ideas about stopping coronavirus. And he was able to test this idea. And the way he does this, I'm not showing you all of the data. I'm just kind of very kind of simplified version of the assay that he's done. And so what you see on the left here is a dish of cells that look, you probably, I don't know how well you can see them, probably not very well, but what you see is kind of a pink sheet of cells. And that means the cells have grown all over the plate. They're very healthy, they're fit, they're happy. And what happens when you infect them with this new coronavirus, SARS-CoV-2, is that you can see there's a very dramatic change in the way that they look. There's far less cells there. They start to clump together in these kind of big aggregates. And that's a sign that they're really not doing very well, and that they're going to die. And so this is called a cytopathic effect assay. And so the assay that Kenneth did was very simple. He simply took the cells and added my aptamer, and then added the virus to see whether the aptamer was able to block the effects of the virus. And hopefully you can see here, even though you can't see the individual cells, you can see that the cells where the aptamer was used in combination with the virus look very much like the healthy cells without the virus. And this and other assays that he's done shows us very strongly that the nucleolin aptamer is able to block the effects of this new coronavirus, at least in this cell-based assay. And so here's where we are right now. We have this experimental drug. We know that from our cancer clinical trials done several years ago that it appears to be very safe for use in humans. And that's a really big advantage. Everybody is hoping that we will get a vaccine for this virus very soon, but we know that that's going to take probably at least a year before that's available to everyone, even assuming that it does work at all. And in the meantime, the only way we can get a drug to patients quickly enough is to use a drug that's either already approved for use in humans, or a drug that's already been shown to be safe in humans. And so that's what we have in this case. And I say, because we're based in Louisville, we were able to do these tests and show that it has activity against the SARS-CoV-2 virus. And so what comes next, I've been fortunate to receive a gift from UPS, and that is allowing us to do additional cell and tissue-based studies to look at the mechanism, see exactly which part of the virus life cycle this is blocking, which will be important as we go to the FDA and try to explain to them why we think this will work. We may do additional studies in animal models of the new coronavirus, and then we'll go to the FDA and seek authorization for human studies, and ultimately conduct a clinical trial in patients who have the new coronavirus. And probably that would be in patients who were sick in the hospital but hadn't yet got very severe disease. And the hope would be then it would stop the disease from getting worse, and those patients could leave the hospital much earlier. And so we're optimistic that that can happen in the next few months, and kind of tide us over until a vaccine becomes available if it works. I'm related to this Aptamer. I've also become involved in the general COVID-19 research going on at the University of Louisville and in Kentucky. And what we're hoping to do is to use the infrastructure that we've built for helping researchers turn their ideas and discoveries into products, and apply it specifically to this kinetic COVID-19 pipeline. So we're keeping our fingers crossed that the grant application we've submitted will come through for that one. And so that's it from me. I'll be happy to take any questions. Feel free to contact me here. You can find out more about kinetic and coronavirus in general, research that's going on at U of L, and how you can support U of L on these web links here. So thank you for having me, and as I say, I'll be happy to take questions.
Great. Paula, thank you very, very much. That was a terrific presentation. It's really interesting to know that this kind of work and research is being done right here in Louisville, and that it's interesting to me, I didn't realize that we had one of the 12 or 13 regional biocontainment units here. We don't think of that sort of thing very often, but that's good to know. I don't see any questions in the pipeline, so this will be last chance. If any members have a question, please type it into the chat room very quickly. And it looks like we do have one. Let's see. Okay. Larry Sloan, I don't see your question. I just see that you want to ask a question. Okay, anybody else have a question? Here we go. Okay, yeah, now they're coming in.
Sure.
Okay.
So I have a question from Julie Smith, which is, am I also involved with antibody testing? Personally, I'm not involved with antibody testing, but the University of Louisville is very heavily involved. We have been, before a lot of these tests became available, we were helping to make sure there was both the molecular testing, which is to see if you have a current infection with coronavirus, and also now helping to develop antibody testing, which will help us determine the prevalence of infection in the community, and potentially can be used to tell if individuals have had coronavirus. And so I see other questions. Oh, Melissa, hi Melissa. Melissa recognizes me from our old collaboration. What else do we have? How long does FDA approval usually take? Well, one of the, I guess, silver linings that has come out of coronavirus is everything is literally moving at warp speed in terms of drug development. So typically, to develop a drug from kind of discovering the effect to getting it approved for human use, it takes years, even decades even sometimes. And so these days, things are happening much faster. People have been, when people have good evidence like we have, we're hoping to be able to do it, change that kind of years of approval time down to months. So we're hoping to start a clinical trial probably this winter.
Okay.
Who would produce this drug once approved for the masses? Well, someone has to produce it before it's actually approved for the masses. We are working with a company right now. It's not finalized, so I can't say too much about it until it is. But they are hoping to do the first clinical trial here at University of Louisville. So that's one of the big advantages of being at the forefront of doing these kind of things. What is the chance we will have a vaccine? No vaccine for AIDS has been developed. I think people generally, again, I'm not really an expert on coronavirus, but my understanding of the situation is people are generally much more optimistic for a vaccine compared to HIV. So HIV mutates very fast, and it's proven impossible to develop a vaccine. The coronavirus is a different type of virus. There's evidence that many people who've been infected with coronavirus do develop antibodies that then can appear to, at least in our model systems, be able to block infection of cells with the virus. So I'm very optimistic that there will be a vaccine, and things are moving very fast. There are already many vaccines in clinical trials, and several of them have shown very good preliminary results. So I think that we will have a vaccine. There are still many questions about how long the protection would last. Whether it would be like a flu type of vaccine where you'd have to have a new one every few years or so. It's not clear yet, but I'm optimistic there will eventually be a vaccine, and we hope this will be something to be used in the meantime. Where does the majority of the grant funding come from? Well, so far, for this particular coronavirus project of mine, our funding has come from the University of Louisville. I'm sure Kenneth Palmer was the one who did the research, so I'm sure he has funding sources that he should be thankful for. I know the Jewish Heritage Foundation has given some for our general efforts. I have funding, just recently received a gift from UPS. And I also have funding just generally from federal agencies such as the National Institutes of Health. Even if we create a vaccine that works, would we likely still see people getting sick and dying? Similar to the flu shot, thousands still die from flu every year. That's true. And again, that's another reason why we should be doing all of these things in parallel, trying to get a vaccine, trying to develop tests, trying to develop drugs, because there's no one thing that's going to fix everything. And so, like you say, we may get a vaccine that works for most people, but maybe it won't work for some people. Or maybe there'll be people who for some reason can't get a vaccine and will need a drug. So yeah, we're hopeful that there'll be a place for all of these things.
Okay, Paula, that looks like the last question. I want to thank you again for a terrific presentation, and also recognize what Mike Kuhl wrote. He didn't ask a question, but he said, thank you for making something so complex easy to understand. And I would echo that. So we do appreciate that.
You are very welcome. Thank you all.
Okay, we're going to wrap up here. 2 or 3 quick reminders. First of all, speaking of Mike Kuhl, he also sent a message through the chat room to let me know and let all of us know that the Anastasia performance has been canceled for this year. So if you were planning on going to that or thinking about it, that will not be held. Secondly, just a quick reminder, if you're interested in engaging on the community issues and some of the things that have been going on over the past week, I would love to hear any ideas that you have. Please feel free to shoot me an email or give me a call. And then lastly, next week, if you're interested in joining in a private chat room for a few minutes before our regular meeting, plan to log on about 11:30 a.m. Watch your email for information from Alyssa as to how to do that. And with that, we are adjourned. Thank you.